Genetics, my favorite subject in the whole world. I think it's powerful evidence for creation. I love talking about this because I think we have got something very, very, very exciting to talk about. Modern genetics, though, gives us three big tools that we can use. First is gene sequencing. You can take a person's genome, which I'll define. Wait, let me back up a second. We got to make a deal. I'm going to have to use some words that most people don't get. Now, this crowd, probably a lot of you do, but some of you aren't going to know the terminology. So if I use a word, that's some $5 highfalutin word that you don't have any idea what that means, and I didn't tell you what it means, you have to tell me. So everybody do this. Ready? Everybody go. Okay, thank you. If I hear that, I'll stop, I'll back up, and I'll make sure that I define the word, because I don't want to talk over everybody's head. This is really important and really exciting, and honestly, I don't think it's that hard to get. And I've had some homeschool moms telling me that. The first time I gave this talk was at a homeschool. I didn't do that. It was at a homeschool convention, and two of the moms came up and said, "Oh, I actually understood that." It's like, okay, good, that's what I wanted. So here we go. Modern genetics, 3 tools. Gene sequencing. I will define genome in 5 minutes, okay? But I have to start off somewhere. Gene sequencing gives us the ability to take a piece of DNA and just decode it. So the human genome is 3 billion letters. We now have all 3 billion letters decoded. Actually, no, I'm sure. About 80% of those 3 billion letters, I have them right here on my laptop. Got a chimpanzee genome here. I've got a whole lot of different genetic information right here on my laptop that all came from gene sequencing. The second tool is genetic manipulation, or genetic engineering. And this is a scary topic. That's not the focus of this talk, but I could talk a long time on genetic engineering and my pros and cons on it. And the third and most important for tonight's discussion is that modern genetics gives us the ability to test history. And that is where the book of Genesis comes in. Okay, so tool number 1, gene sequencing. You've heard of the Human Genome Project. It took over a decade. $3 billion. That's more than $1 per letter that they got sequenced. That's passé, though, because today, in fact, this slide's already out of date. There was a company that a couple of weeks ago ordered enough equipment to sequence 3 human genomes every single day. They're talking about the $1,000 genome, where you can take a cheek swab, send it to a laboratory, and get a DVD, 3 gigabytes of your genome for personal genetics within 2 years. The 1,000 Genomes Project has already started. They have chosen 1,000 people from around the world. The first 2 or 3 people have already been sequenced, and they've already been published. And you can go on the internet and you can download their genomes. This is unbelievably exciting and scary, but there's too much information. Today's geneticist, he doesn't work in a laboratory, he works on his computer. The machines, they just spit out unbelievable and vast amounts of information. So today's geneticist is a data analyst, and he works with computer scientists. He doesn't pour gels and inject things into the gels and make the DNA run through the gels. That's what grad students do. In fact, grad students used to do. The technology today, I mean, since I finished my degree in 2003, everything has changed. That's only 6 years ago. Okay, here's a picture of a genome. Literally, a person's genome. In fact, the person from whom this picture was taken was a man. I know that because there's a Y chromosome. At a certain stage in cell division, your chromosomes line up. And you can take a photograph through a microscope of those chromosomes as they're condensed. And then you can cut out the chromosomes and line them up like this. And notice, you've got all these different chromosomes, they come in pairs. Your genome, literally, is an encyclopedia. 3 billion letters. A, T, G, and C repeated 3 billion times. 3 billion letters, but your encyclopedia is broken up into volumes. We call the volumes chromosomes. You have 46 volumes in your genetic encyclopedia. The volumes have chapters. We call the chapters genes. There are 20,000 or so protein coding genes in your genomic encyclopedia. Okay, everybody with me? None of that yet? We're okay? All right. Part number 2 of modern genetics is the ability to manipulate genetics. This is part of my doctoral work. I was blessed to be able to work in a laboratory that was doing genetic engineering, and I'm not saying this is a good thing, okay, but this is what I did for grad school. I took the genes that code for the bright green and the red fluorescent proteins in corals. I cloned them out of corals. I put them in bacteria. I made bright red and bright green bacteria. And then I cloned them out of bacteria and I put them in a piece of DNA that would express in a fish. And working with some other people, I was able to make bright red and bright green fish. Common zebra danios. You might have some in your freshwater aquariums at home. But mine were bright red and bright green. I made a frankenfish. Whew, for better or for worse, but man, right now, right in this point, I could launch into a 3-day discussion on the ethical and moral ramifications of genetic engineering. But